Welcome, my name is Stephanie Young, and I will be sharing with you our poster entitled Feasibility of a Decision Support Software to Aid in the Interpretation of Somatic Mutations Detected in Hematologic Malignancies. Before we start, I wanted to point out that all co-authors are employees of Roche Sequencing Solutions, and the presentation today reflects my professional views and do not necessarily reflect those of my employer. Also, please note the product disclaimers at the bottom of this slide. Over the past several decades, there's been a remarkable evolution of detection methods to help identify the genetic basis of hematologic malignancies. This started in the 1970s with cytogenetics, which helped characterize the Philadelphia chromosome as a translocation between chromosomes 9 and 22. Fast forward to 2008, and we have the first published whole genome of acute myeloid leukemia, followed by several studies in other hematologic cancers enabled by next-generation sequencing, or NGS. All this work in genomic characterization has enabled insights into disease etiology and therapeutic options, and this is reflected in the evolving classification of hematologic malignancies. We see increasing integration of traditional morphologic findings with the genetic data. Even though the costs of a single NGS assay have been decreasing, the number of genes required for comprehensive diagnostic testing continue to grow. As a result, the time and effort needed for post-sequencing steps, such as variant annotation, interpretation, and reporting, have increased substantially. To address this challenge, we explored the feasibility of an NGS tertiary analysis software called Navify Mutation Profiler to provide automated annotation and interpretation of variants in hematologic cancers. In the study, we leveraged publicly available datasets from cBioportal listed in this table. After downloading, we converted these datasets into individual VCF files and lifted the variants from HG19 to HG38. Then we uploaded each case via API to Navify Mutation Profiler for analysis. Navify Mutation Profiler provides information based on a curated knowledge base, which draws upon public evidence for variant annotation, variant content, and drug approvals or recommendations from the sources listed here. To help prioritize the variants in a given case, the software automatically assigns tier classifications. These tier classifications are based on a joint consensus recommendation published by AMP, ASCO, CAP, and ACMG. In this system, Tier 1 and Tier 2 variants are those of strong and potential clinical significance, respectively. Of the 745 cases we analyzed, 53% had at least one variant classified as Tier 1 or 2. If we look at specific cancer types, this ranged from 96% of cases in MPN to 28% of cases in DLBCL. We also compared tier classifications to a public knowledge base, OncoKB. In OncoKB, we only found information for AML, for the genes highlighted in blue. All the tier classifications were consistent except for IDH1 and IDH2, which depended on the region. If we look at examples of two specific variants in acute myeloid leukemia, we see that Navify Mutation Profiler would automatically tier these as 1A or 1B depending on the region. For IDH1, this is because ivosidenib is approved by the US FDA and Taiwan FDA, but has not yet been approved by the EMA for the European Union, NICE for the UK, etc. Same with IDH2. Enasidenib is approved by the US FDA, Australian TGA, and Health Canada. It is also recommended in the NCCN and ESMO guidelines, but not yet approved by the EMA, NICE, and Taiwan FDA. In conclusion, Navify Mutation Profiler can help prioritize NGS variants and streamline the analysis workflow. This study demonstrated the feasibility of using decision support software to automatically curate and report somatic variants in hematologic malignancies identified by NGS. Thank you for listening.